Firstly, thank you so much for giving me the opportunity to talk over here. I have no financial relationships to disclose. For long-term control of CMV in transplant patients, the adaptive immune responses, which include the CD4 and CD8 T-cell responses, are important. When it comes to CMV reactivation and disease, the role of T regulatory cells is not extensively studied, and data suggests that it could play an important role in the development of CMV disease. Studies show that pre-transplant CD8 T-cell response may be able to predict CMV reactivation post-transplant. The objective of this study was to determine whether pre-transplant T-cell responses, which include the CD4 T-cell response, CD8 T-cell response, and Tregs, may be useful for predicting the risk of CMV reactivation and disease following transplantation. So a cohort of 272 pre-transplant CMV seropositive patients were recruited from 5 centers in Switzerland, and all of these patients underwent solid organ transplantation. None of these patients received any antiviral prophylaxis, and all of them had regular virologic monitoring for the first 90 days post-transplant as a part of preemptive strategy. Whole blood samples were collected and PBMCs were isolated. Following that, live and dead staining was performed. The cells were then stimulated for 6 hours with a live virus of town strain with a multiplicity of infection of 0.03. Profeldin A, a protein transport inhibitor, was added in the last 4 hours. Surface staining was then performed for CD3, CD4, CD8, and CD25, and the cells were then fixed. Intercellular staining was then performed for interferon gamma and FOXP3. Frequency of T-cell responses producing interferon gamma and Tregs were analyzed by flow cytometry. The above flow diagram is an example of how T-cell phenotypes were assessed. On the x-axis, there is interferon gamma with increasing intensity. On the y-axis, there is CD4 with increasing intensity. You see a double positive cells, which on quadrant 1, CD4 positive and interferon gamma positive cells. So, in this study, there was 272 pre-transplant seropositive patients, of which 83 of them had CMV viremia, and 189 of them had no CMV viremia for the first 90 days post-transplant. The CMV viremic patients were slightly older, and majority of them underwent kidney and liver transplantations. There were a few other types of organ transplant as well, which was heart and liver pancreas. There was a significant difference in the CMV serostatus in that there was more donor positive, recipient positive in the CMV viremia group, and which was expected. Of the 83 CMV viremic patients, 10 of them had CMV disease. We first analyzed a positive T-cell response as evidenced by interferon gamma production in the CMV viremic patients and CMV disease patients. In the CMV viremia group, we saw that 72% of the patients had a positive CD8 interferon gamma response when stimulated with live virus, while it was 75% of the patients had a positive response for CD8 interferon gamma positive in the non-viremic patients, and the results were not significantly different. There was also no difference in the percentage of patients with a positive CD4 interferon gamma positive responses between the two groups. Similarly, there was no difference in the percentage of patients that had positive T-cell responses in the disease versus no disease subgroup, leading to a conclusion that both CD4 and CD8 T-cells may be unable to predict CMV viremia or CMV disease. In the next slide, we analyze CD4 responses in donor seropositive and seronegative subgroups. The y-axis is the frequency of interferon gamma produced by CD4 T-cells. And on the x-axis, there are the viremia and no viremia groups. Each dot represents a single patient. The red line represents the median. You can see that there was no significant difference in either subgroup for the prediction of post-transplant viremia. Similarly, the data is not shown, but there was also no significant difference when we analyzed these frequencies for CMV diseases, leading to a conclusion that it was unable to predict CMV disease also. The next slide shows CD8 responses for the D plus, R plus, and D minus, R plus subgroups. Relative to CD4 responses, the frequencies of CD8 responses is relatively higher. But this was true for both CMV viremic and the no viremia group. Regardless, there was no significant difference between the viremia and the no viremia groups. Similarly, we did not see any difference in CD8 responses to predict CMV disease. Next, we look at the Treg responses. On the y-axis, there is percent Treg frequency, and on the x-axis, there are the viremia and no viremia groups. We saw that the patients with CMV viremia had a higher baseline percent Treg frequency compared to the no viremia group, and it was statistically significant with a p-value of 0.035. Patients with a higher baseline percent Treg frequency correlated with the increased risk of CMV reactivation post-transplant. By baseline, I mean that these samples were unstimulated samples. Next, we compared the fold change in Tregs in the viremic and no viremic group when stimulated with live CMV virus of down strain. On the y-axis, there is percent Treg, and on the x-axis, there are the viremic and non-viremic group, which has both stimulated and unstimulated samples. Both the groups, when stimulated with live virus for 6 hours, had a significant decline of Tregs, and it was statistically significant with a p-value of less than 0.001. To conclude, I was able to show that pre-transplant T-cell responses, which include the CD4 T-cell response and CD8 T-cell response, may be unable to predict CMV viremia and CMV disease for the first 90 days post-transplant. Higher baseline percent Treg frequency observed during pre-transplant may be predictive of CMV viremia. Thank you so much.
Thanks very much. This is open for questions. Please
Mike,
actually say your name and the institution. Thank you.
Mike Eisen, Northwestern. So although it's a very small number, but first off, great work. This actually is very reassuring to me. We did something very similar and found similar findings. But the question that I was interested in, although you had a very small number of patients that got lymphocyte depletion, do you have information as to whether or not the T-cell responses were persistent following T-cell depletion? You had 8 in the CMV positive, and I think 10 in the CMV negative group.
Yeah, like I haven't looked into it that much, but yeah, there was definitely a lymphocyte reduction for sure, but I'll have to look into it to know more. Sorry.
Okay, Martina Sester from Hamburg, Germany. I was surprised about the low percentage of individuals that reacted with CD4 T cells especially and CD8 T cells. Have you ever checked this assay in immunocompetent individuals, seropositives?
Yeah, yeah, we did some healthy volunteers as well, and yes, there was a good response. But this you have to see that this is a live virus, so responses would be different. Maybe the MHC class 2 was kind of little bit less for that, so that could be one of the reasons why CD4 responses weren't that great. But yeah, that could be one of the reasons I feel. But yeah, we did see some healthy volunteers in which they were able to express. Yeah.
But was that in a similar percentage range, or
No, it was higher. It was higher CD4 responses actually. Okay. Yeah. So yeah.
Thank you for that presentation. It will be interesting to see whether that difference in Tregs does hold up if you continue to test with a larger group of patients. Because there's a lot of variation between groups, it's possible that the statistical significance could have been chance. That said, I think it's a good approach, and I applaud you for looking at this. I wanted to suggest that perhaps if you looked at secretion of multiple cytokines in response to stimulation, you might start to tease out a difference between those who are able to control viremia versus those that go on to develop viremia. And I think that this cohort of patients is a wonderful opportunity to study immunology in the absence of prophylaxis.
So what sort of cytokines would you kind of like suggest?
Like TNF alpha and IL-2.
Oh, okay, thanks.
Yogesh Kudwa, Rochester, Minnesota. Very nice work. So, can you speculate on the on the actual biology of the Tregs dealing with the CMV? Are you suggesting that there is a subset of Tregs now which is involved in being effector against CMV?
Yeah, so as you saw that in the unstimulated samples, the viremic patients had a higher Tregs, which express FOXP3, so CD4 positive, CD25 positive, and FOXP3. So that's definitely something to look into. Maybe the viremic patients had a higher Tregs. Maybe there is some sort of things going on over there.
So did you look at other readouts, for example, CMV lysis by these subsets?
CMV lysate samples? So you mean?
Actually lysosomal activation in these cells when dealing with the CMV, CD107, for example.
Oh, no. I didn't look into that, but for the next project, I'm definitely looking into that as well. Yeah.
Great. Why don't we move on to your next presentation?
Okay.
Thanks very much.
Yeah. So I have no financial relationships to disclose again. So studies show that pre-transplant assessment of immune responses may be able to predict post-transplant CMV reactivation. NK cell responses may play an important role in addition to the CMV-specific CD4 positive and CD8 positive T cells. NK cells can be divided into various subsets based on the intensity of CD56 and the affinity for CD16. CD56 bright cells could be CD16 dim or CD16 negative, and they are known as the potent cytokine producers, example, interferon gamma. And they represent around 10% of the NK cells. Then there is CD56 dim cells, which could be CD16 positive or CD16 negative. They are known for their high cytotoxicity activity, and the presence of CD16 gives it an effector function of antibody-dependent cell-mediated cell cytotoxicity. In a healthy volunteer, almost 85 to 90% of the NK cells are CD56 dim and CD16 positive. Recent studies show that a subset of NK cells, CD56 dim and CD16 positive, that expresses NKG2C, may be implicated as a memory marker very specific for CMV. This NKG2C is an activating receptor and is expanded during CMV infection, especially with CMV seropositive individuals. The receptor can be stimulated by CMV infected cells. So the objective of this study was to assess NK cell phenotypes in a pre-transplant population and to determine whether NK cell phenotypes assessed pre-transplant would help predict the risk of CMV reactivation and disease following transplantation. Again, 272 CMV patients were recruited. They were CMV seropositive from 5 centers in Switzerland, and all of them underwent solid organ transplantation. None of these patients received any antiviral prophylaxis, and all of them had regular virologic monitoring for the first 90 days post-transplant as a part of preemptive strategy. The methods. So over here, whole blood samples were collected, PBMCs were isolated, live and dead staining was performed, the cells were stimulated for 6 hours with a town-strain CMV virus at a multiplicity of infection of 0.03. Profildin A added in the last 4 hours. Surface staining was then performed for CD3, CD56, CD16, and NKG2C, and the cells were then fixed. Intracellular staining was then performed for interferon gamma, and the frequency of NK cell phenotypes producing interferon gamma and expressing NKG2C by flow cytometry was assessed. Above, you see a flow diagram of the NK cell phenotypes. It's an example. So on the x-axis, there is CD16 with increasing intensity. On the y-axis, there is CD56 with increasing intensity. The red box indicates the CD56 bright cells, and this represents around 10% of the NK cells. Then there are and then on the blue box, there are CD56 dim cells, and the cluster of NK cells that you see are CD56 dim and CD16 positive, and this represents around 85 to 90% of the NK cells. So 272 pre-transplant seropositive patients, 83 of them had CMV viremia, 189 no viremia. Majority of them had kidney and liver transplantation. More donor-positive, recipient-positive patients in the CMV viremic group, and 10 of the patients had CMV disease in the viremic group. So The next slide shows how specific NK cell phenotypes are associated with CMV reactivation. The x-axis has the viremic and the non-viremic patients, while the y-axis has a frequency of subset of NK cell, which are CD56 bright and CD16 negative. Each dot represents one patient. Patient who had CMV viremia had a higher expression of CD56 bright and CD16 negative, with a median of 10.1%, compared to no viremia, which was 5.1%. And it was statistically significant with a p-value of 0.008. Usually, this subset of NK cell is elevated in chronic diseased patients, and it relates with our cohort of pre-transplant patients, and because they were suffering from end-stage renal disease and end-stage liver diseases. Next slide shows NKG2C response to CMV stimulation. Y-axis represents percentage of patients who express NKG2C in a subset of NK cell, which is CD56 dim and CD16 positive. We saw that around 62% of the patients showed no change in the expression of NKG2C, while the remaining patients showed upregulation of NKG2C, which was greater than 20% following CMV stimulation, and it was statistically significant with a p-value less than 0.001. Also, we did notice shedding of CD16 in this population, and it was statistically significant with a p-value of 0.016, although the data is not shown for that. Next slide shows how NKG2C plays an important role in CMV reactivation. On the x-axis, you see our D+, R+ patients who are viremia and no viremia. And on the y-axis, you see a subset of NK cells which are CD56 bright and CD16 negative and express NKG2C. An increase in the NKG2C expression in the non-viremic patients was protective against CMV reactivation compared to the viremic patients. The results were statistically significant with a p-value of 0.03. It was also true for CMV disease as well, where higher expression of NKG2C was protective against CMV disease, and it was statistically significant with a p-value of 0.045. The data is not shown for that. We then looked at the interferon gamma production in response to CMV stimulation. On the x-axis, you see are the CMV diseased and no diseased patients, while on the y-axis, you see is a subset of NK cells who are CD56 dim and CD16 positive that produces interferon gamma. In the no disease patients, a robust interferon gamma response in this subset of NK cell was protective against CMV disease, and the results were statistically significant with a p-value of 0.017, but it was not significant against CMV viremia. To conclude, I was able to show that NK cell responses play an important role in controlling CMV replication. Assessment of specific phenotypes should be further evaluated for prediction of CMV reactivation. Changes in the NKG2C receptor expression may occur in response to CMV. The importance of NKG2C phenotype as a potential CMV-specific subset of NK cells could be further evaluated as a biomarker. At the end, I would like to thank my mentors, Dr. Atul Humar and Dr. Dipali Kumar, for their constant support and moral guidance. I would also like to thank Dr. Lisboa, Dr. Ruiz, and Dr. Han for always helping me out. And last but not the least, I would like to thank the Swiss transplant cohort team, Nicholas Mueller. And this is the picture of my lab in Toronto. It's the Mars, not the planet Mars, but medical and related sciences. Thank you so much.
Thank you very much for an excellent second presentation. Congratulations to you and the entire group in Toronto for doing very nice work. This is now open for questions.
Mike Green, Pittsburgh, great work. So I have a question. So you've looked at CD4s, CD8s, Tregs, and now you're looking at NKs.
Right.
Have you put it together and given us a model of how maybe combining the NK data with the Treg data, since those both had a signal of at least statistical significance, to see in a combined model whether you get a stronger predictiveness or not?
Yeah, so this study was basically performed with a 10-color flow cytometry panel, and we looked into every marker, like all the T cell and the NK cell marker, and this was all we saw, that the NK cells were basically kind of like telling you more about CMV reactivation and disease.
No, I understand that, but it seems to me you have results that with a statistician you could sort of say, so you have a positive result for a Treg, you have a positive result maybe for an NK, CD4, and CD8, and if you put all of those data individually for a given patient into a model to see if by running all four, because when you're doing flow, maybe you're doing more than just you may be doing them all at the same time anyhow, that you come up with a result that by using 2 endpoints as opposed to just using one, you actually get a viable predictor.
Yeah, maybe I'll look into that, yeah.
And to expound on Dr. Green's point, you could also include clinical risk factors for CMV disease in your risk prediction model, along with all these assays, so we might be more able to accurately predict CMV disease.
Right, definitely, because there's a multifactorial issues over here, so definitely we would have to look into each and everything. Yeah.
And my question, though, was about the strain of CMV that you use for your stimulation assays for PBMC. Could you speculate if you used the clinical strain of CMV, whether your results would differ?
Yeah, so actually we were thinking about using the clinical strain as well, but at that time we did not have anything available, and it was kind of a little bit, this particular virus was already grown, and we had done a lot of studies because we used town strain 8169, Merlin, and then we decided town strain would be much better to go through with this study. So that's the reason. But maybe for the next study we might do the clinical strain, because that would kind of like replicate the immune system much better.
Yeah, and with the bacterial artificial chromosome, you could reliably reconstitute the clinical
Right.
strain.
Yeah. Thank you.
I'm sorry, in the interest of time, unfortunately, we'll have to move on to the next speaker, but thank you very much, especially for doing back-to-back talks.
Thank you.